Welcome to this deep dive into the source material. I am so incredibly glad you're here joining us today.
Yeah, it is. It's really great to be here. We have a massive topic today.
We really do. Because we are about to embark on a journey into the absolute core of who you are, how you feel, and how your brain actually wires itself. We are going to be talking about serotonin.
Right. Serotonin.
Now, if you were anything like me, or really anyone who has casually consumed pop science over the last 20 or maybe 30 years,
Yeah.
you probably have a very specific and honestly a very cartoonish image of serotonin in your head.
Oh, totally. The classic happy chemical.
Exactly. It's the happy chemical. You see it
Wow.
printed on T-shirts. You see people getting the little molecular diagram tattooed on their wrists, usually right next to dopamine.
Yeah, I mean, the story we are constantly sold, largely through old pharmaceutical commercials and simplified self-help books, is that more serotonin equals more happiness.
And less serotonin equals sadness or depression. It's presented as this simple, straightforward mood juice, like a slider on a mixing board that you just need to push up to feel better.
But as we are about to discover, that pop culture myth is not just a little bit wrong. It is a massive oversimplification that hides one of the most astonishingly complex and elegant systems in the human body.
Today, our mission is to completely dismantle that happy chemical myth. We are going to pull back the curtain and reveal serotonin's true identity as a master architect of the brain and a macro-scale neuromodulator that dictates how entire networks of your brain communicate. We are treating you as the third person in the room here, someone who is insanely curious about how the machinery inside their own skull actually operates.
To accomplish this, we have assembled a truly massive stack of sources. We aren't just looking at introductory textbooks. We are pulling from a curated selection of peer-reviewed scientific papers spanning from 2003 all the way up to cutting-edge research published in 2024.
And these are heavy-hitting journals.
Absolutely. We are drawing from top-tier journals. We have papers from Nature Neuroscience, Nature Human Behavior, and Nature Communications, as well as foundational reviews from Dialogues in Clinical Neuroscience.
And what becomes immediately apparent when you look at this literature chronologically is a complete paradigm shift in how neuroscientists understand this molecule.
Yes, we are going to explore serotonin's multiple overlapping functions. We will look at how it physically alters the structural changes in your brain, how it governs network connectivity, and how it drives regional activation. We are going to unpack the most up-to-date data, including brain-wide fMRI scans and optogenetic manipulation, to really show you what this molecule is doing at a macroscopic level. It is a story of incredible biological sophistication.
Okay, let's unpack this, starting right at the foundation. Because before we can talk about how serotonin shapes your thoughts and decisions, we have to understand the physical infrastructure. Where is it made? How does it travel?
Right, the plumbing of the system.
Yeah, the plumbing. To build this foundation, we're looking at a highly detailed review paper published in 2010 by Yves Charnay and Lucien Leger in the journal Dialogues in Clinical Neuroscience. The paper is titled Brain Serotonergic Circuitries. And reading this, the very first thing that shifted my perspective was the sheer scale and the physical location of this system. It's not just floating around everywhere all at once.
No, not at all. Charnay and Leger detail that the cell bodies of serotonin-producing neurons, the actual manufacturing plants for the neurotransmitter, are almost exclusively located deep in the brainstem. Specifically, they are clustered in areas called the raphe nuclei, primarily the dorsal and median raphe nuclei.
Which is an evolutionarily ancient part of the brain, right?
Very ancient. It's the kind of rudimentary structure we share with reptiles. Now, to give you a sense of scale, the human brain has roughly 86 billion neurons. The serotonergic neurons in these raphe nuclei number only in the hundreds of thousands.
Wow, so it's a tiny fraction.
It is a microscopic fraction of a percent of your total brain cells. And yet, from this highly localized, deep, primitive location, these neurons send their axons projecting outward to innervate almost every single region of the central nervous system.
They just reach everywhere.
Everywhere. They reach up into the frontal cortex where you do your complex thinking, deep into the basal ganglia, which manages movement and reward, into the hippocampus for memory, the amygdala for fear processing, and all the way down the spinal cord. It is a master regulatory system operating from a very centralized, heavily fortified location.
I was trying to visualize this while reading through the Charnay and Leger paper. The analogy that kept coming to mind was to imagine the raphe nuclei as a central, high-powered water pump located in the basement of a massive, sprawling, multi-thousand-acre farm.
I like that.
Yeah, so that pump represents the relatively tiny cluster of serotonin cell bodies in the brainstem. But attached to that pump is this unimaginably vast and intricate irrigation system. The pipes, the axons, travel miles and miles, branching out to reach every single crop, every field, every greenhouse on the property. Even though the water source is highly centralized and takes up almost no space at all, its influence touches absolutely everything that grows.
That analogy holds up well when you consider the physiological implications.
Mm.
Because those projections reach everywhere, serotonin is implicated in almost every function you can think of. Sleep-wake cycles, memory, cognition, emotion, pain regulation, even your body temperature. But we also need to look at how that water is actually sourced and manufactured.
Right, where does the brain get it?
Serotonin, chemically known as 5-hydroxytryptamine, or 5-HT, requires a specific precursor. That precursor is tryptophan, an essential amino acid.
And essential means we have to eat it.
Exactly. The word essential in biology means our bodies cannot synthesize it. We have to acquire it through our diet from foods like poultry, eggs, cheese, or nuts. Tryptophan circulates in your blood, crosses the highly selective blood-brain barrier, and enters these specific neurons in the morphin nuclei. Once inside, an enzyme called tryptophan hydroxylase 2, or TPOH2, begins the conversion process.
And there's a distinction here between the brain and the gut, isn't there?
There is. Interestingly, TPOH2 is uniquely specific to the brain. There is a different version, TPOH1, which operates in the gut, where the vast majority of the body's serotonin is actually produced to manage digestion. But in the brain, after a two-step enzymatic process driven by TPOH2, you have serotonin ready to be deployed.
And the deployment mechanism is where the irrigation analogy gets a bit of a twist. Because I always learned in high school biology that neurons act like telephone wires. One neuron sends a specific targeted electrical zap to another specific neuron across a tiny gap called a synapse, a direct, private one-to-one message.
That is the classical model, yes.
But the sources point out that serotonin doesn't always play by those strict rules. It has a different way of communicating.
It does. It employs a mechanism called volume neurotransmission, or diffuse transmission, which Charnay and Leger highlight as a defining characteristic of the serotonergic system. While serotonin does form classical targeted synapses, those direct telephone lines you mentioned, a massive amount of serotonergic transmission is non-synaptic.
So it's just released out into the open.
Yes. If we go back to your irrigation pipes, instead of a pipe connecting directly into the root of one specific plant, the pipe has a sprinkler head on it. When serotonin is released from these non-synaptic terminals, it sprays out into the extracellular space. It diffuses over a much wider area, interacting with any compatible receptors in that general vicinity.
Like a mist.
It's like a mist. It acts more like a localized mist or a climate control mechanism for entire clusters of neurons rather than a private text message between two cells. This diffuse spray allows serotonin to act as a macroscale neuromodulator, setting the general tone or the background state of excitability for entire regions of the brain simultaneously.
So if the brain is constantly spraying this serotonin mist to set the tone, there has to be a drain somewhere, right?
Mm-hmm.
If you leave the sprinkler running forever, the field floods, the crops drown, and the system crashes. The paper gets into the deep mechanics of this clearance system, specifically mentioning two proteins, SERT and VMAT2. How does the brain sweep up all this mist once the signal has been sent?
To maintain a highly responsive and dynamic system, the brain needs rigorous millisecond by millisecond control over how long serotonin stays in that extracellular space. The primary vacuum cleaner is the serotonin transporter known as SERT.
SERT, S-E-R-T.
Right. This is a protein located right on the membrane of the serotonergic neuron that originally released the serotonin. SERT actively sweeps the area, pumping the serotonin molecules back inside the cell, clearing the gap so the target neurons can reset.
And then once it's inside,
Once the serotonin is pulled back inside, it needs to be safely stored so it can be used again. Free-floating serotonin inside the cell can be broken down by other enzymes, so it needs protection. This is where VMAT2, the vesicular monoamine transporter 2, comes in. VMAT2 grabs the serotonin and packages it into tiny, durable protective bubbles called vesicles, readying them for the next time the cell fires.
and here's a detail from that 2010 paper that really caught my attention. It's something called vesicular filling synergy. And it completely shatters the idea that neurotransmitters work in total, isolated silos.
It is a phenomenal piece of neurobiology that shows how deeply interconnected these systems are. The researchers found that in certain advanced areas of the brain, like the neocortex and hippocampus, there are serotonergic terminals that actually lack that SERT vacuum cleaner protein.
Which is weird, because how do they clean up?
Exactly. But curiously, they co-express VMAT2 alongside a transporter for a completely different neurotransmitter, Glutamate.
And glutamate is excitatory.
Glutamate is the brain's primary excitatory signal. It's the chemical that makes things happen fast. What they discovered is that the uptake of glutamate into these vesicles actively fosters and accelerates the filling of those same vesicles with serotonin.
That is wild.
It is a chemical synergy. The presence and movement of the excitatory glutamate directly facilitates the packaging and subsequent release of the modulatory serotonin. Serotonin doesn't operate in a vacuum. Its very infrastructure is physically intertwined with the brain's broader excitatory network.
It's just incredible, the sheer mechanical elegance of it all. So we have this foundational map now, the brainstem pump, the vast irrigation network, the volume neurotransmission sprinkler system, and the cleanup crew. But to really understand the gravity of what serotonin is doing, we have to look at what it was doing before you, the listener, were even fully formed. We have to look at the brain in construction.
This represents a vital conceptual shift. We must move from looking at serotonin merely as a chemical messenger in an adult brain to recognizing it as a dynamic structural architect during fetal development.
And to explore this, we turn to a heavily cited 2012 review paper by Klaus-Peter Lesch and Jonas Wader, published in the journal Neuron, titled Serotonin and the Modulation of Neuroplasticity in Networks: Implications for Neurodevelopmental Disorders. We are also layering in insights from a 2003 paper by Patricia Gaspar, Olivier Cases, and Luc Meritot titled The Developmental Role of Serotonin, News from Mouse Molecular Genetics.
Yes, those are foundational texts for this concept.
And this is where the timeline of life on Earth really puts things into perspective. When we think of neurotransmitters, we usually think of them transmitting complex human thoughts, feelings, motor commands. But these papers show that serotonin is phylogenetically ancient. It existed long before brains even existed. It's found in plants, in single-celled organisms, in simple worms.
It's one of the oldest signaling molecules we know of.
Right. And because it has been around for so long, evolution essentially repurposed it to act as a building material, or rather a construction foreman, for the developing vertebrate brain. It isn't just sending messages through the brain today, it actually built the physical roads it currently drives on.
The Gaspar and Lesch papers outline exactly how this works. Serotonin acts as a morphogenetic factor long before classical synapses are even formed in the fetal brain. It guides the physical, spatial construction of neural networks. The papers specifically outline a cascade of events. First, serotonin modulates neurogenesis.
Which is the actual birth of new neurons from stem cells.
Exactly. Then it regulates apoptosis.
Let's pause on apoptosis for a second, because that's a term that sounds a bit counterintuitive when we were talking about growth. Apoptosis is programmed cell death, right? So serotonin is actually telling certain brain cells when to die. Why would a construction foreman want to destroy the building materials?
Because you cannot build a functional, highly efficient structure without clearing away the scaffolding and removing the excess. In fetal development, the brain overproduces neurons. Apoptosis is the necessary, highly coordinated pruning of specific cells to sculpt the brain's final, refined shape. Serotonin helps dictate which cells stay and which ones go.
That makes perfect sense.
Following that pruning, serotonin guides axon branching. It instructs those long communication wires on how to grow, where to turn, and how many times to split. Finally, it influences dendritogenesis, the formation of the receiving branches on the target neurons. The localized gradients of serotonin literally act as chemical breadcrumbs, guiding migrating neurons to their correct final destinations in the developing cortex. It is physically shaping the topography of the brain.
And the implications of that are huge for anyone listening. Because if serotonin is the construction foreman, what happens if the foreman is given the wrong blueprints, or goes on strike, or gets too much overtime during that critical developmental window in the womb or early infancy?
By using molecular genetics, researchers have been able to study genetic models where serotonin homeostasis is transiently altered during fetal development or early postnatal life. And what they have found is profound. These temporary early life fluctuations in serotonin do not just cause a passing phase, they cause permanent, lifelong changes to adult behavior.
It's hardwired in.
They permanently modify the fine wiring of brain connections, particularly in the somatosensory cortex and the prefrontal networks. The architecture is fundamentally built differently, and it remains that way into adulthood.
Which means that your foundational baseline for how you interact with the world, how you handle stress, how you perceive threats, all of that was heavily influenced by the ebb and flow of serotonin before you even took your first breath.
Lesh and Waiter connect this directly to neurodevelopmental disorders. Deficits or dysregulations in early 5-HT synaptic signaling fundamentally impact the development of what they call the biosocial brain. This early wiring dictates how the adult brain will later process contextual fear responses, how it manages emotional learning, and how it handles social cognition.
It sets the baseline frequency.
Exactly. If the serotonergic scaffolding is altered during development, the entire emotional processing network operates on a different frequency for the rest of the organism's life. It highlights that serotonin's role in mental health isn't just about a day-to-day chemical imbalance that can be easily fixed with a pill. It is deeply, inextricably rooted in structural plasticity and how the brain's networks were woven together in the very beginning.
Okay, so serotonin builds the house. But once the house is built and you're an adult moving through the world, how does serotonin control the temperature, the lighting, and the security system? To understand that, we have to look at the receivers, the receptors. If serotonin is the master key, the receptors are the locks, and honestly, there are an overwhelming number of locks.
The sheer diversity of serotonin receptors is exactly what allows a single, simple molecule to have such vastly different and sometimes contradictory effects across the body and brain. For this segment, we are leaning heavily on a deeply insightful 2017 paper by Robin Carhart-Harris and David Nutt, titled Serotonin and Brain Function: A Tale of Two Receptors, as well as a meticulous 2013 paper by Francesc Artigas, titled Serotonin Modulation of Cortical Neurons and Networks.
When I was looking through the Artigas paper, the numbers alone were staggering. We aren't just dealing with a serotonin receptor type A and a serotonin receptor type B. There are 14 different 5-HT receptor subtypes, categorized into 7 distinct families. 14.
It is a massive family of receptors.
That means when that serotonin mist sprays out of the brainstem sprinkler, it can hit 13 completely different types of locks. And every single one of those locks triggers a completely different biochemical reaction inside the receiving cell. It's like having a master key that turns on the lights in the kitchen, locks the front door, starts the washing machine, and sets off the fire alarm, all at the exact same time, depending on which keyhole it enters.
It is the ultimate Swiss Army knife of neuromodulation. But to keep things focused and to really understand the core of human behavioral adaptation, both Artigas and Carhart-Harris zero in on 2 specific receptors that have deeply contrasting cellular functions and behavioral outcomes: the 5-HT1A receptor and the 5-HT2A receptor.
And Artigas focuses on a specific brain region for this, right?
Yes. Artigas focuses his analysis heavily on the medial prefrontal cortex, or MPFC. This is a brain region highly evolved in humans. It is the seat of complex decision-making, emotional regulation, and impulse control. Through detailed electrophysiological recordings, Artigas demonstrates the opposing forces these 2 receptors exert on the exact same networks.
And this is where we have to talk about pyramidal neurons.
Yeah.
Because they are the main actors in the prefrontal cortex.
Yes, pyramidal neurons are the primary excitatory cells of the cerebral cortex. They get their name from their triangular, pyramid-shaped cell bodies. You can think of them as the primary thinkers or the main executive decision-makers in that region. Their long axons project to other areas of the brain to execute commands. Now, when serotonin binds to a 5-HT1A receptor situated on one of these pyramidal neurons in the MPFC, it hyperpolarizes the neuron.
Let's break down hyperpolarization for the listener. Neurons communicate via electrical charges. They have a resting baseline charge, and if that charge gets pushed positive enough, it crosses a threshold and the neuron fires. It sends its signal. Hyperpolarization means the serotonin is pushing the internal electrical charge of the cell, more negative, further away from that firing threshold.
Right, it's inhibitory.
It's slamming on the brakes. It quiets the cell down and makes it much harder to trigger.
And then, situated within the same cortical networks, and sometimes on the very same pyramidal cells, you have the exact opposite happening with the other receptor. When serotonin binds to the 5-HT2A receptor, it depolarizes the neuron.
So it pushes it closer to the threshold.
It makes the inside of the cell more positive, pushing it closer to its firing threshold. It is excitatory. Artigas notes that 5-HT2A activation significantly increases excitatory postsynaptic currents. So you have a single neurotransmitter, serotonin, capable of both slamming on the brakes via 5-HT1A and hitting the gas pedal via 5-HT2A in the most advanced cognitive centers of your brain.
So that is the cellular level, brakes and gas pedals. But what does this actually mean for you as you're going about your day? How does a hyperpolarized pyramidal neuron translate to how you handle a terrible day at work or a fight with your partner? This is where the Carhart-Harris and Nutt paper brings in their bipartite model. This framework totally changed how I view stress. Let's look at how they map these two microscopic receptors onto our massive psychological coping mechanisms.
Carhart Harris and Nutt proposed that these two receptors govern two entirely different, evolutionarily conserved strategies for dealing with stress and adversity. The 5-HT1A receptor pathway is what they term the passive coping pathway. Because its function is largely inhibitory, the breaks you described. Activating this pathway moderates anxiety, blunts the sharp edges of psychological pain, and improves your general stress tolerance.
It's just grinning and bearing it.
It is the brain's default, everyday response to adversity. It helps you keep your head down, grit your teeth, and endure a difficult, unchangeable situation without spiraling into panic. Importantly, this is the primary pathway that is chronically enhanced by traditional SSRI antidepressants. It builds resilience through endurance and emotional blunting.
To put that into a real-world context for the listener, imagine you have a daily commute that takes 2 hours in grueling traffic, or a boss who is just constantly mildly toxic. You can't just quit your job tomorrow. You have to pay rent. The 5-HT1A receptor is what allows you to endure that. It turns down the volume on your frustrations so you can survive the daily grind without having a breakdown. But then you have the 5-HT2A pathway, which is the complete opposite. It's the wild card.
The 5-HT2A pathway mediates what they call active coping. Instead of just enduring the stress and turning down the volume, 5-HT2A activation does something profound. It opens a window of heightened neurological plasticity. It makes the brain's networks more malleable, more sensitive to the environment, and more capable of fundamental reorganization.
It forces a change.
It allows the brain to completely change its outlook, its deeply held beliefs, and its behavior when facing extreme adversity. If the passive coping mechanism fails, or if the environment becomes so hostile that simply enduring it will lead to destruction, the active coping mechanism is engaged to force a radical, highly flexible adaptation to the environment.
The analogy I wrote down for this, and this was my huge aha moment when reading these sources, is to imagine your life as a house, and a massive hurricane is hitting it. The hurricane is the stress, the trauma, the adversity. The 5-HT1A receptor is what helps you endure the storm. It's the psychological equivalent of boarding up the windows, taking shelter in the basement, pulling a blanket over your head, and just riding it out. It keeps you safe and stable while the wind howls.
It helps you survive the immediate threat.
Right. But the 5-HT2A receptor, that's what happens after the storm, when you walk outside and realize the roof is completely gone. You don't just sit in the rubble and try to endure it. The 5-HT2A receptor gives you the flexibility, the profound plasticity to say, okay, the old house didn't survive this. The old way of living is dead. I need to design and rebuild the house differently so it can withstand the next storm. One receptor helps you endure the storm. The other gives you the architectural plasticity to rebuild differently after it passes.
That analogy captures the functional divergence beautifully. It completely reframes serotonin from being a simple happiness chemical to being a highly sophisticated manager of environmental adaptation and survival. It dictates whether you tolerate a stressor or fundamentally change yourself to overcome it.
Okay, so we've looked at the microscopic level, the single neurons, the specific 1A and 2A receptors. But the brain doesn't operate as a bunch of isolated individual cells. It operates in massive brain-wide networks. We need to zoom out. We need to look at how serotonin modulates the entire globe of the brain all at once. And for this, we have some absolutely fascinating, highly technical fMRI data from 2 recent papers.
This is where the story jumps into the macro scale, moving from cellular biology to network neuroscience. We are drawing from a 2022 paper published in Nature Neuroscience by Pier Giorgio Salvin and colleagues, titled Serotonin Regulation of Behavior via Large-Scale Neuromodulation of Serotonin Receptor Networks. And we will follow that with a brilliant 2024 paper by Hamada and colleagues, titled Optogenetic Activation of Dorsal Refi Serotonin Neurons Induces Brain-Wide Activation.
Let's begin with Salvin's methodology, because it is a masterclass in modern computational neuroscience.
It really is. Salvin and his team wanted to know how the distribution of different serotonin receptors influences brain-wide activity and human behavior. To achieve this, they combined human brain transcriptomic maps with resting state fMRI scans.
Let's translate transcriptomic maps for a moment, because it sounds intimidating, but the concept is brilliant. The researchers took post-mortem human brains from people who had donated their bodies to science, and they painstakingly mapped exactly where the genes for all these different serotonin receptors are expressed.
So they created a physical atlas of receptor density.
Exactly. So they end up with a high-resolution 3D atlas showing, there's a heavy density of 5-HT1A receptors in this square millimeter, and a heavy density of 5-HTTC receptors over in this region. Then they laid that 3D genetic receptor map over top of functional MRI scans from living humans who were just resting in the scanner. The fMRI shows which areas of the living brain are naturally talking to each other, firing in sync. By fusing these two massive datasets, they discovered what they call SRNs, serotonin receptor networks.
They mathematically extracted the functional connectivity patterns that were uniquely associated with the spatial distribution of specific serotonin receptors. The major finding of the paper is that it is not just the presence of serotonin that matters. It is the specific admixtures, the unique combinations and ratios of different receptor types in different brain areas that dictate human behavior across these large-scale networks.
And to link this to actual behavior, they used a specific statistical tool.
To definitively link these biological SRNs to actual human psychological traits, they used a sophisticated statistical technique called canonical correlation analysis, or CCA.
To picture how CCA works, imagine you have two massive spreadsheets open on your computer. Spreadsheet A has all the brain scan data showing the strength and connectivity of these serotonin receptor networks for hundreds of people. Spreadsheet B has all the behavioral data for those exact same people. How impulsive they are, how depressed they feel, how they process rewards, their personality quiz results. CCA is an algorithm that looks at both massive spreadsheets simultaneously and finds the hidden mathematical bridges between them.
It looks for predictive patterns.
It essentially asks, what specific pattern of connectivity in the brain data perfectly predicts a specific pattern of behavior in the psychological data?
And through this canonical correlation analysis, Salvin's team discovered two entirely independent modes of population variation in humans. Two distinct behavioral profiles driven by different serotonin receptor networks. Mode 1 links specific SRNs, notably those involving the 1A and 2C receptors, to what they term waiting impulsivity and antisocial problems. This is the dimension of behavior control, aggression, and the ability to delay gratification.
And what about mode 2?
Mode 2 was completely independent. It linked different SRN patterns, primarily involving the 2A and 1B receptors, to a negative bias in reward processing, major depression, and feelings of panic.
Which perfectly aligns with the historical paradox of serotonin. For decades, scientists have argued in the literature, is serotonin about controlling impulsivity and aggression, or is it about processing negative emotions and depression? And the Salvin paper essentially says it's both. It just depends on which specific receptor network you are looking at. It's like finding out that the volume knob and the tuning dial on a radio are controlled by the same hand, but they do completely different things depending on which circuit they are wired to.
It resolves a decade-old debate by mapping the behavior to the network rather than just the molecule. But Salvin's work in humans, while groundbreaking, is ultimately correlational. They observed patterns. To prove causality, to prove that serotonin actually causes these brain-wide network changes, we must turn to the 2024 paper by Hamada. To prove causality, you have to manipulate the system directly, and Hamada achieved this using optogenetics in mice.
Optogenetics is a technique that is genuinely revolutionizing neuroscience. For the listener who might not be familiar, scientists literally genetically engineer specific neurons, in this case, the serotonin-producing neurons, deep in the dorsal raphe nucleus of mice, to become sensitive to light.
No, these channel rhodopsins.
Right, they insert proteins called channel rhodopsins, which are borrowed from light-sensitive algae. Then they implant a microscopic fiber optic cable directly into the mouse's brainstem. When they shine a specific wavelength of laser light down that cable, it instantly forces those, and only those, targeted serotonin neurons to fire. It is a literal on-off switch for serotonin, controlled entirely by light.
It provides unprecedented temporal and spatial precision. You are no longer relying on messy systemic drugs. Hamada and colleagues took these optogenetically modified mice and placed them inside an ultra-high field 11.7 Tesla MRI scanner.
Which is incredibly powerful.
Very powerful. To give some context, a standard hospital MRI is usually 1.5 or 3 Tesla. An 11.7 Tesla scanner produces an incredibly powerful magnetic field, allowing for breathtakingly detailed functional imaging of the tiny mouse brain. But here is the critical methodological leap that sets this paper apart. They performed these fMRI scans while the mice were completely awake.
Why is that a leap?
Previous fMRI studies using optogenetics on serotonin neurons had almost exclusively been done on anesthetized mice. And historically, when you activate the dorsal graphy in an anesthetized mouse, you see widespread suppression of cortical activity. The whole brain essentially quiets down. This older data reinforced the assumption that serotonin is primarily a global suppressor.
But when Hamada turned the laser on while the mice were awake, they saw the exact opposite, right? The brain didn't quiet down at all.
Activating the dorsal raphe serotonin neurons in awake mice induced powerful brain-wide activation across both cortical and subcortical areas. The frontal cortex, the basal ganglia, the ventral tegmental area all lit up with coordinated activity. This completely challenges those older assumptions. It demonstrates unequivocally how state-dependent serotonin networks truly are. The exact same chemical release from the exact same neurons causes global suppression during the anesthetized unconscious state, but drives widespread, highly coordinated activation during wakefulness.
So the background context matters just as much as the chemical itself.
It proves that serotonin isn't just a simple static switch. Its effects are entirely dependent on the existing network dynamics, the ambient environment, and the conscious state of the organism.
It's like trying to judge what a conductor does by watching them wave a baton in an empty concert hall versus a full orchestra pit. The physical movement of the baton is the exact same, but the output silence versus a symphony is entirely dependent on who is awake and ready to play.
We have now established the anatomy, the developmental scaffolding, the opposing receptor functions, and the brain-wide macroscale network activations.
Which brings us to the climax of our deep dive. We are taking all of this biology, the reprompting nuclei, the 1A and 2A receptors, the massive brain-wide networks, and we are bringing it into real-time, high-stakes human scenarios. We are looking at live-action serotonin in human decision-making. We have two breathtaking papers from 2024 to guide us here. The first is by Seth Batten and colleagues, published in Nature Human Behavior, titled Dopamine and Serotonin in Human Substantia Nigra Track Social Context and Value Signals During Economic Exchange. The second is by Michael Colwell and colleagues in Nature Communications, titled Direct Serotonin Release in Humans Shapes Aversive Learning and Inhibition. Let's set the scene for the Batten paper, because the methodology is intense.
It is a remarkable clinical setup.
They had human patients who were awake, in an operating room, undergoing deep brain stimulation surgery for Parkinson's disease. Parkinson's patients often undergo surgery to implant electrodes to help control their tremors, and they are kept awake so the surgeons can test their motor responses. While the neurosurgeons had electrodes inserted deep into a region of the basal ganglia called the substantia nigra, the researchers had the patients play a game on a screen. The game is called the ultimatum game.
The ultimatum game is a classic economic behavioral task used extensively to study social decision-making, fairness, and punishment. In the game, a proposer offers to split a sum of money with the patient, who acts as the responder. Let's say the proposer has $10. They can offer a fair split, $5 and $5, or they can offer an unfair split, keeping $9 and offering the patient $1. The catch is, if the patient rejects the offer because they feel it is unfair, neither person gets any money.
It's a test of whether you'll punish someone, even if it costs you.
Exactly. Furthermore, Batten introduced a critical variable. The patients were explicitly told that some offers came from real human beings in another room, while other offers were generated randomly by a computer algorithm. While the patients considered, accepted, or rejected these offers, the electrodes in their brain were taking sub-second measurements of both dopamine and serotonin levels simultaneously.
And to measure that, they used a technique called fast-scan cyclic voltammetry. Now, to picture that, imagine dropping a microscopic chemical sensor directly into the brain that acts like a high-speed camera. Instead of taking a picture of the brain's anatomy every few seconds like an MRI, this probe rapidly changes its voltage to cause neurotransmitters nearby to oxidize or lose electrons. It measures the exact electrical current produced by that oxidation, allowing researchers to track the exact number of serotonin and dopamine molecules flooding the area hundreds of times per second.
Real-time neurochemistry.
It lets them watch the neurochemicals fluctuate in real time, millisecond by millisecond, as the social interaction unfolds. So what did they find? Because culturally, we always hear dopamine and serotonin mentioned together as the twin pillars of mood. How did they differ when the patient was deciding whether to accept a fair or unfair offer from a human versus a computer?
The results brilliantly uncouple these two neuromodulators, showing how distinctly they govern behavior. The researchers found that dopamine acted exactly as advanced prediction error models suggest. It tracked the social context. The dopamine signal spiked differently when the patient was dealing with a human compared to a computer. Dopamine cared deeply about the social dynamic, the intent, and the who of the interaction.
And serotonin.
Serotonin, however, behaved entirely differently. Serotonin purely tracked the current offer value. It didn't care if the offer came from a human who was trying to cheat them or a computer that was just generating random numbers. It provided a generalized, rapid, objective signal representing the raw value of the offer on the table. It is a moment-to-moment value tracker devoid of the complex social bias that dopamine carries.
So if you get a terrible offer, dopamine is whispering, hey, this guy is disrespecting you. He's trying to rip you off. Punish him by rejecting it. While serotonin is just holding a calculator saying, well, the offer is only $2, but $2 is still mathematically greater than $0. It's incredible to see them working in tandem but doing such fundamentally different jobs at a sub-second level.
Now, let's move to the final piece of the puzzle, the Colwell 2024 paper. This paper is special because of how they manipulated serotonin. Most human studies use SSRIs, like Prozac or Lexapro, but studying acute serotonin release with SSRIs is incredibly tricky.
Because of the autoreceptors. Right. They block the reuptake vacuum cleaner, yes, but they rely on the neuron actually firing to have an effect. And if you recall, neurons have built-in thermostats called autoreceptors.
Yes, the autoreceptor is a crucial negative feedback mechanism. When an SSRI blocks a reuptake, serotonin initially builds up in the synapse, but the presynaptic neuron has autoreceptors, specifically 5-HT1A receptors, on its own cell body. When these autoreceptors detect that the serotonin level is getting too high, they send a signal to the neuron to stop firing entirely.
Like a thermostat shutting off a furnace.
It's exactly like a thermostat detecting that the room is too hot and shutting off the furnace. This is why SSRIs can initially decrease serotonin firing and why they often take weeks to show clinical benefits, as those autoreceptors slowly desensitize. Colwell, however, wanted to bypass this slow, complicated thermostat entirely.
So to bypass it, Colwell utilized a drug called fenfluramine. Fenfluramine is a selective serotonin-releasing agent, or SSRA. Unlike an SSRI, which merely blocks the vacuum cleaner and waits for the cell to fire, an SSRA actively forces the issue. It enters the neuron, goes straight to those VMA2 vesicles we talked about earlier, and forces them to dump their stored serotonin directly into the synaptic cleft, regardless of what the autoreceptor thermostat is saying.
It forces a massive release.
It causes a robust, immediate, and direct flood of synaptic serotonin. This has allowed the researchers to study the unadulterated effects of a massive serotonin spike in healthy humans. They administered this drug and then put the participants through a rigorous battery of computational behavioral tasks.
And the behavioral outcomes they documented perfectly synthesize everything we have discussed today regarding passive endurance, active coping, and network modulation. What happens when you flood the human brain with serotonin? The effects were highly specific and deeply tied to how we handle negative, unpredictable environments. First, they found that increased serotonin significantly reduced the participants' sensitivity to aversive loss outcomes.
Meaning they didn't get as upset by losing.
Correct. When they were punished or lost money in a probabilistic learning task, the serotonin spike essentially shielded them from the psychological sting of that negative feedback. They didn't overreact to the loss. They didn't panic and immediately change their strategy just because of one bad outcome. Secondly, it drastically enhanced behavioral inhibition. In tasks designed to measure impulsivity, the participants became much more cautious. They were able to hit the brakes and control their impulses, especially during tasks that involved aversive emotional interference.
So bringing this all back to you, the listener, if we tie Colwell's findings back to Carhart-Harris and the concept of coping, we see the entire system working perfectly to protect the organism. Picture yourself in a high-stress scenario, maybe getting a harsh performance review at work. In that stressful, aversive context, a spike in serotonin biases you toward impulse control. It forces you to stop and think, rather than reacting blindly or defensively. And simultaneously, it acts as a biological shield, reducing your sensitivity to the sting of that negative feedback so you can actually process the information being given to you without being overwhelmed by fear, anger, or loss. It is the ultimate biological shock absorber.
The Colwell study provides the definitive, live-action proof in humans of the theories we explored from the earlier neurobiology papers. It shows the neurochemical in action, mediating our relationship with an unpredictable, stressful, and often punishing environment. It allows us to pause, evaluate the objective value of the situation as seen in the Batten paper, and either endure it or initiate the plasticity required to change.
Let's recap this monumental journey. We started deep in the basement of the brainstem, looking at the Drophin nuclei acting as a massive centralized water pump, using volume neurotransmission to act as a sprinkler system, setting the tone over the entire brain. We saw how, before you were even born, this ancient molecule acted as a construction foreman, literally sculpting the physical architecture of your fetal brain networks through neurogenesis and pruning.
It built the roads.
We explored the incredible tapestry of receptors, focusing on the dual forces of the 5-HT1A and 5-HT2A receptors, the passive endurance to weather the storm, and the active plasticity to rebuild the house differently. We zoomed out to the macro scale, seeing how unique combinations of these receptors create brain-wide networks that govern everything from waiting impulsivity to negative bias, and how the state of being awake totally changes serotonin's global impact. And finally, we saw it operating in real time, tracking the objective value of a social exchange in the substantia nigra and acting as a biological shield against aversive loss and impulsive reactions.
It is a profound departure from the simplistic happy chemical narrative. Serotonin is the master regulator of how your brain interfaces with reality, particularly when that reality is challenging. And this brings me to a final thought, directed right at you. We have seen today that serotonin isn't just regulating your mood, it is regulating your brain's physical plasticity and its threshold for change via those 5-HT2A networks. We know that extreme stress or adversity can trigger the system to open a window of profound neurological adaptation. Therefore, if serotonin opens the window for profound neurological adaptation when we face adversity, the question isn't just how to artificially increase your serotonin levels to feel better. The real question is, what new environments, what new challenging behaviors, or what new perspectives are you actively exposing yourself to while that window of plasticity is open?
Oh, that is such a powerful thought to end on. Because plasticity just means change. It doesn't guarantee the change is good unless you direct it. Thank you so much for joining us on this incredibly deep dive into the source material today. We hope you walk away looking at that little molecular diagram on your coffee mug with a whole new level of awe and respect for the machinery inside your head. Keep questioning, keep exploring, and we will see you next time.